To fabricate a good tool device in biomedical engineering, we need to model the systems, all elements of a system. And in this subject, using an electrode for recording EXG signals from the body, we have a system consists of electrode and a skin, and we call this system the electrode skin interface. And in the next slides we will model the interaction between the electrode and the skin using some data from the previous slides, and we will add some new information about the skin and the interaction between the layers. And after that we have a complete electrode skin interface model. As you can see in this slide, let me plug the pen. As you can see in this slide, a typical anatomy of the skin has three main layers: the outer epidermis and dermis and subcutaneous layer, the deepest layer of the skin. The outer layer of the skin, epidermis, consists of some layers, and the outer layer of the epiderm is stratum cornea. This is the hydrated layer, and it means that the conduction of this layer is very low. But the deeper layers, dermis and subcutaneous layer, have some ducts or vessels full of some liquid, some electrolyte, and therefore the conduction of the deeper layer is better than the outer layer. I repeat it, the conduction of the outer layer is very low because the outer layer, stratum corneum of the epiderm, is a dehydrated layer. Therefore, the ions inside this layer cannot be moved easily, freely. Therefore, the conduction of this layer is very low. And the deeper layer, because of its vessels and sweet ducts in these layers, the conduction is good. Now we place the electrode on the skin. And we have a model for the electrode from the previous page. As you can see, we place exactly the same model, electric lamp model here. These elements for the electrode and RS for the gel or electrolyte. And also we have a voltage source here for modeling the half-cell potential. And EHE is for half cell of electrolyte. H half cell, E electrode, electrolyte. Okay. And now here we have a layer, epidermis. Epidermis, as I mentioned before, is a dehydrated layer. Therefore, it acts like dielectric, like a high resistance layer, and we can use a model we presented before for the electrolyte here, because we have a ion storage at the two sides of epidermis. Therefore, we have a capacitance and also a resistance for the epidermis and a resistant Ru for subcutaneous fully full of ion layer, and a SE voltage source for modeling the skin electrode voltage. A skin electrode. Now our model is completed. But as you can see, we have another part in this model connected by dashed lines. As I told you before in the previous slide, we have some ducts and vessels inside dermis and subcutaneous layers. But the difference between the vessel and the sweet ducts is reaching the outer parts of the skin. The sweet ducts are passed from the subcutaneous dermis and also epidermis, and we have the entrance and we have the holes of the ducts on the outer layer of our skin of the ducts, but the vessels, the capillary vessels, only only located in the dermis, not epidermis. And let me let me say this: the blood in the vessel and the sweat in this duct are a good conductive path, but the vessels only is under the epidermis layer, and the vessels cannot pass through the epidermis, but the sweat ducts can pass through the epidermis, and the sweat can reach to the outer layer of epidermis. When we are recording EXG, ECG, EEG, EOG, and something like this, especially ECG, EEG, EMG, we only use the reaction and the capability of the vessels to transfer ionic current, and we record the ionic current provided by the vessels. But in recording GSR, galvanic skin response, or EDA, electrodermal activity, we will monitor the performance of the brain in controlling the stress, or for example, for lie detection and something like this. We use GSR and EDA in some cognitive research and cognitive studies. And as you can see, we need these ducts. We need the performance of the sweat glands while we is under stress, or for example in bad situation, when lying, fear maybe leads to some sweat. Therefore, in GSR and EDA, we need the performance of these ducts, including some sweat inside it, and sweat is a solid liquid, and solid liquid has a low impedance. Therefore, the impedance of the epiderm will be reduced. Therefore, our model needs another part for modeling this sweat ducts and sweat glands performance, and this part is for modeling the performance of the sweat gland and sweat ducts in recordings like GSR and EDA. Okay. And also you can read the text in this slide by yourself. Based on the structure of the electrodes, we have some problems, and one of the most important problems in recording is motion artifact. As you can read, when polarizable electrode is contact with an electrolyte, a double layer of charge forms at the interface. As I show you in the previous slide. Movement of the electrode will disturb the distribution of the charge and results in a momentary change in the half cell potential until equilibrium is reached again. We have a steady state in the structure of the electrode, and this double layer of charge will fix if we don't have any movement. But movement of the electrode leads to disturb the distribution, and the distribution of charge, the changing in the distribution of the charge leads to a change in half cell potential, and therefore this half cell potential will affect the output or the recorded signal by this electrode. Motion artifact is less minimal for non-polarizable electrode. AgCl electrodes have less motion artifact compared to metal electrodes or polarizable electrodes. Signal due to motion has low frequency, so it can be filtered out when measuring a biological signal of high frequency components such as EMG or axon action potential, AAP. However, for ECG, EEG, and EOG, whose frequencies are low, it's recommended to use non-polarizable electrode to avoid signals due to motion artifact. For example, when we record ECG, we want to have a stable you can see the signal moves up and down. We call this line base line, and the baseline wandering or baseline changing, moving will occur because of the motion artifact. We want to have a fixed ECG, fixed baseline in ECG. For solve or for overcome this problem, we should use a good adhesive connection to skin. We use good adhesive electrodes, and we should fix it. We should clean the skin and use some electrodes. We call them floating electrodes. But the main solution is using a fixed electrode, good electrode with good adhesive electrode.